Hello, my name is Professor Shitij Kapur, and this is episode 3 of Life Beyond Coronavirus, the Expert View. Today, we're exploring the race for a vaccine. What does a vaccine look like? Who will get there first, and why does that matter? What does it take to bring a vaccine to the market? And what are the politics of distributing a life-saving vaccine during a global emergency? To look at these and other issues, I'm joined by 4 world-leading experts located right here at the University of Melbourne. Professor Sharon Lewin is the inaugural director of the Peter Doherty Institute. She's an infectious diseases expert and a basic scientist. Professor Kanta Subbarao is the director of the WHO Collaborating Center for Reference and Research on Influenza at the Doherty Institute. And Professor Damien Purcell is the head of molecular virology at the Peter Doherty Institute. Professor Chris Edmond is an expert in international economics in the Faculty of Business and Economics. Now, Sharon, as a world leader in infectious diseases, what is the likelihood of getting a vaccine?
I'm quite optimistic we will have a vaccine. What I don't know is when we'll have it and how effective it's going to be.
There you go. So you actually escaped the question of the likelihood, but we'll come back to you on that. Kanta, you were involved in the SARS vaccine. What have we learned from that helps us now?
So we learned 3 really important things. We learned what the principles would be that would underlie a successful vaccine. We learned that neutralizing antibody protects, and we learned what the target on the virus is to induce that neutralizing antibody.
That's right. Those are 2 things. What's
Those are 2
the
things.
third?
The third is that we also learned what safety signals we need to watch for in developing a coronavirus vaccine.
All right. Damien, you're developing the vaccine now. What do you think is our biggest challenge?
I think the biggest challenge is feasibility. We need speed, and that actually limits the choices you have available if you want to make it available in a rapid manner.
Well, we'll come back and learn more about it. Chris, as an economist, what do you think of when you think about a vaccine for this virus?
So I guess I'm most thinking about what the possible bottlenecks on the supply side are, like what we need to do to kind of be ready to produce at scale very quickly if and when we have a suitable vaccine, and then also the distribution within and across countries.
Well, we'll come back to learn more about that, but let's start perhaps with where we are today with COVID in Australia. If we could just look at the number of new infections and the epidemic and the pandemic as it is going through the country. Sharon, what's your take on where we are?
We are in a very good place in Australia. We've at around 7,000 cases, under 100 deaths, and less than 20 cases on average each day around the country, other than localised outbreaks.
Now, I think on the 22nd of last month, I might have the date wrong. We were down to 4 cases. So if you look at that, it came really low. Why didn't it just go away?
Well, we're always going to have some level of infection in the community until we have a very prolonged time without any infections. So normally if you're looking for eradication of an infectious disease from a community, you would wait for 2 incubation periods, which is about a month in the coronavirus world. So we were down, and we never got to 0, we got to these very low numbers. Now the minute we lift social distancing restrictions, you increase, and we still have only done that very modestly, you increase your likelihood of being exposed to another person that's carrying the virus. Right. And I think that's why we will still, as we slowly go through these 3 steps of lifting restrictions, we will almost certainly see new infections. But the hope is, and the strategy is, that these will largely be localised outbreaks that can be dealt with strategies around testing, isolation and quarantine.
So if we keep it this low, do we really need a vaccine?
We absolutely need a vaccine. To keep it this low is actually quite a heroic effort.
Right.
You just look in our health departments, the numbers of people that have been employed to create this army of contact tracers. You look at the testing that we're doing. We've actually done a total of almost 1 million tests in Australia, And some states are doing 20,000 odd tests a day. This is enormous effort to maintain where we are.
That's right.
Plus, our lives are far from normal,
Indeed.
even as restrictions lift. So I think we definitely need a vaccine.
Now, of course, your normal life was not about the coronaviruses. You are a world expert on HIV. What have we learned from HIV vaccine? Do we have one for HIV as yet?
We don't have an HIV vaccine. We have invested enormous amounts of money in a vaccine and learnt an enormous amount. And it's interesting that many of the strategies being tested right now for coronavirus come from those years and decades of investment in HIV. So there's no doubt that investment has been valuable. We have not got a vaccine though, and there's a lot of reasons why we don't have a vaccine for HIV. HIV is not coronavirus.
All right. Well, let's learn from something that comes closer to coronavirus, and that's, of course, SARS. And Kanta, you were at the heart of the SARS and the Ebola efforts to make a vaccine. So tell us for the SARS, the first SARS, this is the second SARS, what happened with the vaccine in the first SARS?
Yeah, so that was really interesting. I mean, there were enormous efforts to develop vaccines. Again, like today, a number of different platforms were used. And there was a lot of development, lots of preclinical development. Only 2 vaccines made it into clinical trials. They were both very small phase 1 clinical trials. So we did learn a lot from the preclinical testing as well as the 2 phase 1 clinical trials. But SARS went away. It didn't come back.
So how long did it take in SARS-1 to get the first human trial started, the phase 1?
Right. So that was it was very quick, but the outbreak was brought under control with public health measures in about 4 months.
Even quicker.
Yes. And so the vaccine developers basically had to make a decision about whether to proceed with vaccine development and vaccine production or not. And a lot of those, what are called go, no-go decisions, were based on whether the virus re-emerged.
So for SARS-1, the companies started, they got to a point, but they never followed through and completely documented a successful SARS-1 vaccine.
That's correct. And in fact, if we had, we'd be even better placed today because we would have had more experience with a vaccine going into large numbers of people.
So that didn't help the epidemic because the epidemic went away.
That's right.
What's the likelihood this epidemic's going to go away?
Oh, I think this is completely different. This is not going to go away. So the big difference is that SARS-1 was caused by repeated introductions from animals into people.
Right.
There was relatively little transmission from person to person.
Right.
It required very close contact. And so it was possible, and also people were very severely ill. So it was possible to identify cases and trace contacts and stop those chains of transmission. We can't do that with SARS-2 or COVID-19 because there's a lot of asymptomatic infection. There's a lot of person-to-person spread. It's very efficiently spread.
Right.
And so it's hard to identify every case and to limit every single, do all the contact tracing necessary.
All right, so it seems we will need a vaccine. Damien, you are always working on vaccines. For those of us who are not experts in vaccines, tell us what a vaccine actually is. All of us have been vaccinated, but what is a vaccine? How does it work?
Well, basically, a vaccine is taking a component of a virus or a bacterium and then presenting it to a patient, a candidate, in a way that enables their immune system to grapple with that and come up with a protective immune response.
So why doesn't it make them sick if it's getting to get an immune response?
Because often our immunity drives some of the pathogenesis. And what Kanta was just saying before with the SARS vaccine is evident in several other vaccines, that the antibodies in particular sometimes can actually carry something like a virus to a place in the body where it normally won't go.
Right.
So in the case of the SARS coronavirus, some of the negative antibodies from some of the vaccines carry the virus perhaps in macrophages to places in the body where it drives additional disease.
So it could be a bad thing. So some vaccines could lead to more complications.
Well, this is what Kanta was referring to as a negative signal for safety. So there are 2 kinds of antibodies that we've recognised with the SARS type of vaccines and other related viruses. Neutralising antibodies that bind onto the tip of the spike in particular and prevent that sort of spring-loaded machinery from allowing the virus to enter cells.
Right.
But then there's non-neutralising antibodies that bind to other parts of the virus that don't actually inactivate its capacity to infect a cell.
Right.
And those antibodies potentially can be a problem.
So, all right. So, because, generally the world thinks antibodies is a good thing, and if you've got antibodies, you've got perfect immune protection. That's not the case. Now, there's also these two terms one hears about passive immunity and active immunity. What's the difference?
So, active immunity is, as I've said before, presenting the antigen, and your immune response develops and learns, it's educated to make an effective neutralising antibody response.
Right.
A passive vaccination is actually harvesting antibodies either in convalescent plasma and delivering those passively to a patient, or more recently, monoclonal antibodies that are highly targeted to the tip of the spike, for example, the bit that is the neutralising, what we call epitope, and very potently inactivating that one component.
Right. So the active immunization is when you make the antibodies, and passive is when you get it from outside somewhere and give it to you.
Yes.
Does the passive work as well?
So with the convalescent plasma, which is something that is the only sort of passive vaccination experiment done so far, it can be effective, but it's been very difficult to understand the components, the kind of antibodies within those samples.
Right.
But the monoclonal antibodies, there's actually a very interesting trial in HIV underway just now where they've made a very potent monoclonal antibody and assessing whether it can passively protect patients, immunise patients.
All right, so now that we
find out.
understand the sort of basics, what were you going to say, Sharon?
I was going to say we've got a history of using passive vaccination, which we use, say, with hepatitis A. After an outbreak of hepatitis A, you can give people antibodies to protect them from becoming infected. Damien mentioned HIV. Ebola potentially has that effect. Rabies, you can use that.
So we have a long history of doing it.
Yeah, and the question of whether it will work in coronavirus, we don't know. In influenza, it doesn't work terribly well. But in other infections, there's that principle that you can...
Right.
The downside of it is it doesn't last long. It only lasts for the period that you have the antibody on board, which is probably about 6 weeks. Maybe it could go out to 3 months.
You're making vaccination very complicated here. I thought it was simple. You got the vaccination, you never get the disease, but obviously things are complicated. Of course, the complicated aspect is developing. So now, Damien, you had a wonderful graphic that you were going to show us about the different stages, because we keep hearing all of these estimates of people who say a vaccine would be developed in 18 weeks, it was at some point in time, and now more reasonable people are saying 12 to 18 months. Why does it take this long? Can you just walk us through the different steps of it?
So usually that question is complicated because it depends on who you ask. If you ask the people in the laboratory setting, how long does it take you to make the vaccine? It's usually months. So that's the preclinical stage of development. So preclinical is actually coming up with something that is a candidate.
Right, so that's the preclinical testing as we can see on the screen. And then what happens once the people in the lab have said, yes, take it to patients?
So then it enters the late stage preclinical, which is, it has to actually undergo the first round of toxicology testing, usually in 2 species of animals.
All right.
And then produced at sufficient quality grade to go into a phase 1 trial in people to understand.
And what do you test in the phase 1?
So phase 1 is all about safety. So will this vaccine, we've tested it in 2 animal species, it's demonstrated safety there. When taken into phase 1 into people, a limited number of people, is it also going to demonstrate safety, not cause any unknown response?
So let's say it's safe. What happens next?
So next is more complicated because now you enter into the logistics phase of manufacturing at higher quantity and at GMP level, so it's something that has to pass a lot of safety considerations for other reasons. And then it's delivered to more people where it's tested for immunogenicity and the first signs of efficacy.
So immunogenicity means you're just looking for whether it makes the right kind of antibodies and signals like that.
Correct. Is it making the neutralising antibodies, for example, or what's the level of non-neutralising antibodies, detrimental antibodies?
And when you've done that, it goes into those big phase 3 trials.
And then it's at phase 3 when it's tested in hundreds of thousands of people, ideally, in a setting where there's risk of transmission.
Right, so if we had a vaccine today, we don't have hundreds of thousands of people to test in Australia. Where would you test it?
You test it where they're in the midst of high incidence of infection,
Right.
which is of course happening still in parts of Europe, likely in Asia and Africa.
So you hope they're still there when the vaccine gets there.
Exactly.
Otherwise, we'll have the same SARS-1 problem. We got the vaccine, but the disease was gone. But how many of these vaccines are in trials at the moment?
Well, there are 10 vaccines in clinical trials currently.
Right.
Roughly half of those in China, half of those outside of China.
Right.
There's about over 100.
So 100 in
the
different
early
stages
phase.
and 10 now into humans. Now, Chris, what would be the best models for financing this work?
So generally, we think of kind of two alternatives, or two kind of extreme. One is like a kind of patent or licensing type model, where you think about rewarding a successful innovator with a kind of a monopoly, subsequent monopoly profits over the successful vaccination. The other is kind of more of a public-spirited model, where you think about offering what's referred to as a prize, a kind of essentially a lump sum that then allows the public sector, say the government, to essentially nationalize the drug. And then you do that when you think either that the public good benefits of the drug are so large, which is presumably the case here, that you want to not worry about the kind of the monopoly pricing that you might have on some narrower
All right.
Treatment.
So it's either a patent or a prize, but government in the public interest often requisitions things. So when they want to build a big grand central station, they just go and requisition, all the little shops that were there and build a grand central station. There couldn't be something more important for the nation and the world now. Why, once it is successful, couldn't the government go and just requisition it and provide a fair return and then take it over?
Well, there's a couple of reasons. So one is that it's very difficult to requisition intellectual property in that sense, right? That it's once the actual understanding of the vaccine has developed enough, it's very difficult to kind of you can requisition land because you can essentially have an exclusive use of that land. It's very difficult to have exclusive use of the intellectual property. The other is that if you're thinking about countries like Australia or other countries where the public sector is already extensively involved in funding research, a lot of that research has clauses that basically make it possible for the government to be involved in the subsequent development of the drugs. And so it's very difficult contractually to requisition in the same way that you might requisition land.
Now we know there's hundreds of these around the world. There's 10 in trials. Let's say one of these becomes successful. And we're hearing all of these things, how governments engage in bidding wars behind, and then tests by the millions are flown in on planes. Let's say if someone came to Prime Minister Scott Morrison and said, I've got a vaccine, it really works. And of course, the right thing that Scott Morrison should do is should consult our experts in health and say, look, does it really work? And let's say Sharon, Kanta, and Damien say, yes, Prime Minister, this works. And he turns to you and Chris say, how much should I pay for this? So how much should Australia pay for a vaccine? Just give us a sense of the number. A vaccine that could come today, that could work in Australia as good as conventional vaccines usually do.
So I have a very simple round number for you. It's $1 trillion.
Wow. It is round and it's simple, but it sounds very big. How did you come to $1 trillion?
So that number is, if you think of the Australian population of 25 million, if you think of a 1% fatality rate, and if you use the Australian government's standard value of a statistical life of $4 million, then that, if you multiply that out, that's $1 trillion. So I can make that number much bigger or much smaller depending on which of the
it's big enough already. Now tell me.
So if you use,
go ahead.
So if you use, for example, the U.S. value of the statistical life that's used in U.S. government decision making, which is more like $11 million US, you would double or triple that number. But if you thought that the fatality rate was much lower than 1%, then you would lower it. But then if you want to take into account all of the other implications of COVID over and above mortality, then that would increase the number. So like 1 trillion is a nice round number.
So you think 1 trillion is about the fair value of what this vaccine could mean to us. Put that in some perspective. I mean, how long do we as a nation have to work to make $1 trillion?
Half a year.
I'm sorry? Half a year?
Half a year.
So it's 6 months.
That's national income for 6 months.
So that's what the value of it would be. 6 months of your work without pay
For everybody.
for every single person in this country. All right. Now, if he did, let's say the Prime Minister somehow found the money and got us to pay for it, which is exactly what it means, and it did come to Australia, Sharon, who would we give it to first in Australia? I mean, he's bought it for all of Australia, and in the fullness of time, everyone will get it, but it'll come in batches, as these things do. Who would you give it to first?
Yeah, it's a tricky question which many people are thinking about, and there's two things to think through. Where would it have the biggest impact, and what's the acceptability of those decisions? And those decisions can't just be made by scientists saying, or clinicians saying, you know, old people get the sickest, we give it to old people first. You need to think about the way the virus circulates in the community, the effectiveness in different populations, and where you'll have the biggest impact. And at the same time, you also need to engage the public, because whenever you have a resource in short supply, and you're going to make some tough decisions, you always should make them in partnership with the people it's going to affect. And in fact, this is some work that we've been doing in the lead up to, in what we call preparedness research, through a network that I lead, funded by the NHMRC, called APRISE, where some of our investigators, ethicists from the University of Sydney and Newcastle, and involving also our staff from University of Melbourne, Jodie McVernon, have been running these citizens' juries about about what to do if all this work has largely been done in flu. In pandemic flu, you get a new vaccine, who gets it first? In flu, it's a well-worn dialogue of people who get sickest and are at greatest risk. Usually would be the people that get it first. But to start talking to communities so they understand that there may well be a hierarchy. So there's some really interesting work being done by Jodie and Angus Dawson and Peter Massey and others, looking at acceptability of how you roll out something that we may not have unlimited supply of. In the coronavirus world, I would say that you'd be most likely wanting to reduce transmission in the general community, rather than necessarily targeting people that are most likely to get sick, because the elderly are less likely to make a good response to the vaccine.
So they don't respond. Which brings me to the other question. Of course, I've made it an Australian question just for us to get some sense of perspective. This is in reality a global question. And you, of course, can't think about it in the WHO perspective. We've had one experience before with H1N1, with the swine flu pandemic. And there was a vaccine for it finally. How did we decide how to distribute that, and who got that first?
So at the time, I wasn't involved at the WHO level, but it is, you know, as Sharon said, the sort of organization of influenza discussions is really very mature, and so countries have had pandemic response plans, and they've had consultations. They also have established, many of the large governments have established relationships with the vaccine manufacturers to purchase vaccine or to stockpile it. All of that would be new with the coronavirus vaccine. And what would be important to figure out is whether individual countries might place restrictions on companies that are on their shores to provide for domestic use before they're allowed to distribute widely.
Wow.
That had been dealt with back in 1976, we thought, for influenza, but it might be a completely new discussion again with coronavirus vaccines.
Right. And given, if I heard you correctly, you said half of the trials are happening in China, or these are China-developed vaccines. The other half seem to be the US-developed vaccines.
And some in Europe.
And some in Europe.
Australia.
It would be...
In Australia.
Now, of course, it would be most interesting if the Australian vaccine got there first, but you never know. But Chris, thinking on the fly, you said that the Australians should be willing to give up 6 months of their national income for a vaccine like this. Let's say we use the same standard for everyone in the world, and the world gave up 6 months of its income. What price would you put to that?
That's an astronomical number, but it's like, I don't know, something on the order of $100 trillion. But you wouldn't do it that way, right? I mean, I think that the framing things as like giving up 6 months of your income is making that calculation seem like too tough for the average person to bear, right? So the whole point of a government is to think of issuing debt in that situation and smoothing that expense out over a long period, over like a generation, over 30 years or more, and paying that, thinking of that as like something you pay off like 1% or 2% per year.
All right.
And that's quite reasonable. And so the actual headline number is very, very large, but that's not the burden. That's not the sort of like the true economic burden of the development.
But that could land up making some company fabulously rich, whichever way it comes about. But coming to the previous examples we've had of these exceptionally costly drugs that have came around, HIV was a good example. But in the end, we were able to find innovative mechanisms such that in large parts of the world, and now we're doing that with the hepatitis C drugs. So how did that work? How did we go from a very market-oriented calculation to a much more social and global aspect?
First of all, I think that is happening and will happen in the current environment.
Good.
The story in HIV was driven by global community advocacy. So in the late 90s, we had effective drugs. They were largely available in high-income countries. They, on average, cost around in the order of $20,000 to $30,000 a year, and the drugs that were being used were on patent, because all companies have to make returns on the investments they put in to develop a product. In the early 2000s, when it became apparent that that the global epidemic was way beyond high-income countries and a huge need in low and middle-income countries, there was an agreement that low and middle-income countries could access generic drugs while they were still on patent.
Right.
That led to massive manufacturing of generic drugs, primarily out of India, and an adjustment of costs, which still exists today, so that if you have HIV in India, it would cost you $800 a year for drugs. If you have HIV in Australia, it costs you it costs our government $25,000 a year. And the same principle, a fantastic outcome of global advocacy, actually, and equity. And now about 60% of the world living with HIV can access drugs. I think we do have similar sorts of programs for vaccines right now. We have global organisations like Gavi that ensure access to vaccines across the world, Gates, the WHO, a range of global organisations that are actually talking right now, those same players, about how vaccine access to low and middle income countries will come about.
Now that's heartening. So we understand the biology, we understand how we work out the money, you say the ethics are in place, but in the end you got to make 7 billion of these. So give us a sense of how many times the current production rates of vaccines this is. I mean, how many vaccines do we usually produce a year to give to all the children of the world, etc.? This is...
I can't give you a
Kanta might know
number.
from a flu perspective, because every year you need to manufacture
How much flu vaccine do we make every year?
So we do make millions and millions of doses of influenza vaccine, but there are large parts of the world that don't use influenza vaccine.
Right.
So it's very patchy. There are, huge parts of the world, including much of China, that doesn't use influenza vaccine. So it's not a great model to base it on.
But I'm still thinking, even if it's millions and millions, let's give it 100 million.
The US alone
giving everyone
more.
in the world is 70 times or, huge. So, Damien, what are the challenges for just producing these things?
So vaccines in general are very bespoke products. They're usually produced by a purpose-built factory. And this is why I think feasibility is a crucial question. So some of the candidates that are coming through now, like the genetic-based vaccines, RNA vaccines, are untested for their scalability and probably unable to produce millions and millions of doses, as you're asking about. So I think the existing capacity with more generic kind of vaccines, so protein subunit vaccines, as University of Queensland are producing, are capable of actually delivering at least tens of millions of doses within Australia.
Right, and of course Australia has been a leader in that, and it was one of the first sort of vaccines that was off the block. So where is the UQ vaccine sitting at the moment?
So currently it's made in a sufficient quantity to test in animals. Right. It's undergoing or finished testing in mice, and Kanta has tested that vaccine. So
Kanta, you might want to tell us more about it.
Well, we've tested the immunogenicity, that is the immune response in the mice to the vaccine, and that looks very promising. And now it's in, so mice that we test, laboratory mice, are inbred, and so you get a very consistent response in every mouse that you administer the vaccine to. So the next step is to test it in animals that are outbred.
Right.
And you also want to be able to challenge those animals with the SARS virus to see if it works.
Right.
So those studies are going on right now.
So that's where it stands. And let's say all of that works out. Does it next go to humans?
Yes, so the next phase would be a phase 1 study in humans. That would be a small study in groups of people that would receive the vaccine, and we would look for safety. And of course, because it's being done in a small group of people, we can also look at the immune response. But the primary focus of phase 1 studies is safety.
Right. Now, of course, all of this, again, assumes vaccines just work. But of course, what you pointed out is that they're complicated and they don't work for everyone. So Kanta, the influenza vaccine, what % protection does it actually provide?
So that's a complicated question because our flu vaccines actually contain 4 strains.
Okay.
And we do very well with 3 of the 4 in terms of effectiveness, in terms of effectiveness of protection. So the way we measure effectiveness of the influenza vaccine is a study design that's generally agreed upon internationally. And so in a good year, we'll get vaccine effectiveness in the 60 to 70% range. But
that's a good year.
It's a good year. But one of our components is quite problematic, and that's the H3N2. And we have seen very low effectiveness in certain years. So it's still better than not being vaccinated. And 3 of the 4 components do very well.
Right. But so it's not a panacea.
No.
But all of this is assuming that the virus stands still. Now, of course, one thing we learned from flu is that the virus keeps, genetically moving, or at least...
So we don't anticipate that will be a big problem with coronaviruses. They do not change genetically at the pace that influenza viruses do.
Right.
And, we're watching, millions of cases now, and there's an unprecedented amount of genetic sequencing being done in real time. And so we have to see what immune pressure does to a coronavirus. We have never been in this situation before. We haven't been in a situation of vaccinating people with a coronavirus vaccine and looking at whether the virus tries to escape that immune pressure. Right. So those are things that are still unknowns. I don't anticipate it will ever be the level of antigenic change that we see with flu, because these viruses behave differently biologically. But we also have not put this virus under that pressure.
To test. And how long do these immunities last? Because many of these childhood vaccinations we take them once in childhood and then never worry about them, or 2 or 3 times in childhood and never worry about it.
So we've learned a lot over the years. We used to think most vaccines given in childhood lasted a lifetime, and we've actually found that they don't. And we have to boost either in adolescence or later in adulthood. So I think that when we start off with vaccine development, we really are not thinking that it's going to be lifelong. And we need to do the studies to see how long vaccine-induced immunity lasts. I think if natural infection gives you lifelong immunity, there's good hope that a vaccine would also give you lifelong immunity. But coronaviruses might actually be able to reinfect you during your lifetime. And so those are data that we still need to develop. But the expectation would be to protect for a certain number of years, and you would follow people that are vaccinated to look at the longevity of the immune response and essentially take your cues from that as to whether it's time to revaccinate.
Right. Now, I'm turning back to you, Chris. We gave you a very simple example. Someone came with a perfect vaccine, offered it to the Prime Minister to start tomorrow. We're realizing the world is unlikely to be like that. Vaccines are complex. They take time to produce and manufacture. In the meantime, of course, our economy is being impacted. Australia is lucky that, as I think Premier Daniel Andrews said, we can start gradually returning over a long period of time. What impact, how would you measure, and what's your sense of what impact it's having, and how quickly could it restitute before we got the vaccine?
Okay, so the the shock to the economy from the need to achieve social distancing is unprecedentedly both large and sudden. So we've never seen ever a shock to the labour market that's happened so quickly. And so it's the speed of that shock that I think has most economists concerned, because we've seen big recessions before, and I think we know a fair amount about how to deal with big recessions. The politics of that is different, but we know technically how to deal with them. But what we've never seen is a collapse in the U.S. labour market of the severity that we have seen, of the suddenness. So that speed of the onset and like what that does is something that's really new under the sun.
Now,
it's not.
Yeah. Go ahead.
No, it's fine. Go ahead.
No, I was going to say, looking at the numbers though, and you would know the numbers much better than I do. I thought it was 30 million plus U.S. jobs lost, which is practically almost 3 times as many jobs as Australia has in the first place. The impact on jobs in Australia seems to have been lesser. Am I correct in that? And if that's the case,
No.
why?
No. So we're going to the Australian Bureau of Statistics will release its labour force survey today, and we'll get the first real data on the Australian labour market today, in about 2 hours' time. So we maybe are getting slightly better labour market outcomes than is true in the US, but that difference so far is probably marginal. So, the unemployment rate is like a tricky number to think about here, because many of the labour market implications are not actually through people becoming unemployed because they're being stood down and not actually being counted in the unemployment statistics, at least not yet. So much of it is coming through like drastically reduced hours and things of that kind and other disruption. So you kind of need a kind of a broader set of like what's going on in the labour market. But as I was saying a minute ago, it's not so much that the extent of unemployment is going to be unprecedentedly large, although it will be very large, but it's the severity with which that arrived. So normally it takes 6 months to a year for unemployment in a recession to peak. And we've probably seen it peak in like 6 weeks.
And is that a good thing or a bad thing?
The speed of that, that's a terrible thing.
Oh, okay. I thought maybe we got it, if it's quickly in, it could be quickly done. People keep talking about these weeks.
Well, maybe, right? So that is the optimistic interpretation. And so the real uncertainty here is how much extra disruption is done by that speed, by the suddenness. So if it was true that the dynamics on the other side are very similar to the way we go in, which is that's just like a big hypothesis that's unproven. But if it was true, then yes, that's an optimistic scenario. But I mean that's a hope.
Okay. Well, it seems there are hypotheses on the biological side, and then there are hypotheses. We're living in a world of speculation and hypotheses. What about this hypothesis? When could we know that there isn't going to be a vaccine?
I will start with
Yes.
that. I think we will keep going and going till we get a coronavirus vaccine. We've seen this sort of severe coronavirus outbreak happen 3 times now, all from species jump. We know these coronaviruses are in bats. We're likely to see another. I think we've learnt that we will need to keep going here, finding antivirals and a vaccine. And I think a good example is HIV, where 30 years later we still don't have a vaccine, and yet about 2/3 of the National Institutes of Health budget for HIV is still focused on a vaccine. We will need a vaccine to end an infectious disease such as HIV, or in preparation for another coronavirus. So I think what we will do, though, as a global community is have to adapt and think of ways we're going to live with coronavirus, whether it's with antivirals, testing, social distancing, immunological track records of exposure. There'll be a whole lot of other ways we'll live with coronavirus. But I think the scientific effort for finding a vaccine has to continue. And we also learnt that with Ebola, I should add, that there were some vaccines in partial development and therapeutics back in 2014 when we had the biggest Ebola outbreak after. And then things did change, and we actually do now have an Ebola vaccine.
Okay. So I agree. Scientifically, we've just got to keep at it for very good reasons that you point out. But, pragmatically, people need to plan. And what I mean by that is, with what is happening now with social distancing and return, all our current offices need to be totally remodeled. On the other hand, if I knew a vaccine could potentially come in 12 to 18 months, I wouldn't take down the CBD and build it again. I would just find ways of working for a given amount of period. So what do you think is the peak expectation time after which we need to go to a plan B? How long should we keep our hopes up before we realize, well, maybe not, let's find another plan?
Well, I think about a year to a year and a half of watching to see where some of these really exciting developments take us. There are 10 vaccines that are in clinical trial now, and there are a number of, hundreds of vaccines that are in preclinical development. And I think we will see interesting data from them, and we'll get some idea of whether they work when they get into people. I think in the end, if any of these work, or if all of them work, we'll need every drop of every type of vaccine. Right. It's not going to be one platform alone, because as you said, we need to have vaccine for 7 billion people. So I think that I'm optimistic, very optimistic, and I think that but we will have to be patient.
But I'm getting a window. How long do we have to hold our breath, even though the science may well take 30 years?
Yeah, I think a year or two.
So that timeline would be reasonable to see all of these efforts mature. Now, of course, people will start hearing about it, and the first thing they'll hear about a successful vaccine will merely be success in phase 1. Right. So after a success in phase 1, generally, what is the percentage success rate that you'll actually go all the way and get a successful vaccine?
So it's not 100% for sure. It's a lower number, about 40%. I mean, it's hard to say with this coronavirus vaccine.
Right.
I'm, like Kanta, optimistic, but I think it's not going to be a single one-size-fits-all solution. We might actually have a successful vaccine, but it might be an effective vaccine in the elderly, for example, and not so good in children. So
you're saying even if there is a very successful phase 1, right? Don't call your victory as yet.
Don't throw it away, because there's a difference between age groups. There's also immune histories in different communities that might actually be relevant. So I think we need many, many candidates. We're going to have to keep testing them, maybe multiple different populations potentially, to come up with the right solutions.
Now, we spoke about vaccines today, but of course people are developing treatments, and even though there are 100 vaccines in development, there are, I think, over 300 registered different clinical trials. How could that work out, Sharon, that treatments might get there before the vaccine does?
Well, antivirals or treatments for COVID-19 are really important, and they could have a big impact on the whole scenario, because if you had an antiviral that stopped people getting very sick and going into intensive care, you release that bottleneck. If you have an antiviral that stops people even going to hospital, even better. And you might even have an antiviral that could stop transmission or protect people from becoming infected. So antivirals could play a role. Most of the antivirals being evaluated currently are largely repurposed drugs. They're not designer antivirals for COVID-19. They will take longer to develop.
They'll take longer to develop. Well, it's been wonderful to learn about the virus and the vaccine, and I read somewhere an author who wrote, he says, we will learn a lot about the virus, but we will learn more about ourselves. Ourselves as people, ourselves as community, ourselves as nations. So my question to all of you, and I'll take each of you in turn. In all of this, we'll learn some good things about ourselves and some bad things about ourselves. I'll leave the bad aside. So tell me, what have you seen, or what can you see emerging in all of this that has strengthened your faith in humanity? Sharon, I'll start with you first, because I've always been starting with you first. So, and Chris, just so that you're prepared, the economists can have the last word on what has strengthened your faith in humanity.
Well, I think we've seen that humanity is highly adaptable. We use the word resilient a lot, but I think it's true. And what's given me, recently, I'm incredibly proud of what's happened in Australia. Australians Scientists have worked really effectively with government. We've had bipartisan support for having an evidence-driven policy. And then the community have really responded with social distancing. It's extraordinary what we've achieved in Australia. We do public health incredibly well here.
And may that long continue. And indeed, the world is looking at us in New Zealand, places like that with envy.
Yeah. And all in 8 weeks, 12 weeks' time for people to get to grips with this strange new virus, to trust the scientists, to trust government decisions. It's been pretty extraordinary.
Wonderful. Kanta, what helps your faith in humanity through all of this?
Again, it's a word that Sharon just used, and that's resilience. I think it is amazing. The other thing that I'm really happy to see is how much technology and technological advances have translated very rapidly into digestible, usable information and how it's influencing care, diagnostics, therapeutics, vaccine development. It's all very rapid. This is just 4 months
That's
old.
remarkable. I was reading this morning, there have been about 53,000 publications, scientific publications, on coronavirus. In a world where usually it used to take us 3 months to get any paper even published.
Exactly.
So clearly a lot has changed, and you're right. Damien, what?
Well, absolutely amplify Sharon's statement about the Australian community response in particular, but also the government response, listening to scientists, being asked advice and being listened to is actually very refreshing for a scientist. But also among the science community, I think that there's been a real difference in the way we've tackled this particular problem. We've worked together. We've realised that it's a race against the virus, not against each other. So there's a lot of collaboration. We're not trying to compete with University of Queensland, for example. We're trying to synergise with them.
Right.
There's a lot of crosstalk and assistance between not just scientists in Australia, but internationally, also with pharmaceutical companies, WHO. So there's a lot of hope there for collective effort really getting to the solution in the fastest possible time.
That's right. In fact, Tom Friedman in the New York Times wrote a beautiful editorial saying, this is just the warm-up for the real dialogue we need to have with nature, and that's about climate change. But we'll come to that perhaps on another day. Chris, what have you seen in all of this that enhances your faith in humanity?
So I'm going to pick up on both those. So exactly amplifying everybody else, just the response of the Australian community, the Australian scientific and medical establishments, and the government response has really given me a lot of encouragement. And exactly on the climate change front, because it's exactly what we need there. And maybe that has not been as productive a dialogue in the past, but maybe this experience gives us a kind of a paradigm to follow on that front too.
Wonderful. Well, look, we've always been a lucky country, and may that long continue. But of course, it wasn't all luck. I think we are fortunate that all elements of our society and community came together. So with that, it is time to bring this discussion to a close. So please join me in thanking and even applauding, if you can, our experts. And let me thank you for your interest. This has been episode 3 of Life Beyond Coronavirus, the Expert View. The next 3 episodes will continue to explore the impact of COVID-19 on our world, looking past the present and looking out to the future. And we're looking for your suggestions. What things do you think will change in life beyond COVID? We encourage you to share your ideas and questions on the University of Melbourne's social media channels, and the details of which are, of course, on the screen now. We will try to address your suggestions with expertise and with evidence. Till next time, thank you for joining us. Good day.